Hello, today we will discuss about the HIV virus. It can cause the AIDS, acquired immunodeficiency syndrome. In this way, the immune system will be damaged due to the CD4 damage. So here let's begin to understand, for example, the immune system first of all, for understanding the HIV virus infection. So here is the antigen representing cell which that is recognized by the toll-like receptor to the pathogen, and the pathogen can be virus, bacteria and protein. And after the recognition will bind with the MHC class 1 molecule with the CD4 receptor and this T cell receptor of the CD4, and the CD4 molecule will be bound to recognize this antigen by the piece of antigen representation. So after this antigen, first of all, the dendritic cell produce or antigen representing cell produce the cytokines. The cytokines for attraction of the CD4 cells. So when the CD4 cell will be attract after the recognition by the showing with the MSC class 1 molecule with a piece of antigen, and this piece of antigen will be recognized and the CD4 will produce the cytokines too. And in this way the innate response will be occurred. The recruitment of the neutrophil, mast cell and macrophage, and on the other hand the basophil. So this is the innate immune response, while the adaptive immune response is also important for the long-term production of the antibodies from the B memory cell, which that is the B memory cell will be produced from the naive B cell, and the quick time at the time of infection, the plasma cell is also produced by the proliferation of the naive B cell, and this naive B cell produces the plasma cell and the B memory cell. Plasma cell is produced at the response of the pathogen, and this plasma cell produce the antibodies. These antibodies is the immunoglobulin, and this immunoglobulin will collapse the pathogen and engulf by the innate response, that is the neutrophil engulf and macrophage. So it will decrease the um, uh, uh, clear the infection. So in this way you can see, and this is the B memory cell and the plasma cell we produce. But let's begin to understand the HIV. So first of all we will discuss about the transmission of the HIV. So here is a chart which that is showing the contamination of syringes which that is cause the HIV, and sexual contact and vaginal semen and exchange of body fluid, as well as the breast milk through mother, can cause the AIDS if the carrier mother AIDS acquired immunodeficiency syndrome will be occurred due to the loss of the CD4 we will discuss now. So now here is I am drawing the CD4 cell which contain a CD4 molecules and T cell receptor. So let's begin to understand the CD4 molecule which that is here. You can see that is the cross section from the here we will discuss about. So there are many CD4 molecules are present, but here I am showing for understanding just one. So in this way here is the virus first of all we will discuss. This virus contain a RNA virus, and RNA virus two-stranded RNA virus, and it's contain a reverse transcriptase enzyme, integrase enzyme and the protease enzyme, and this enzyme is used for the replication into the host, into the CD4 cell. So here is the CD4 molecule as well as CCR5 and CXCR4. This is a CCR5 or CXCR4 is basically also recognized, and this here is a virus contain a glycoprotein spikes, and this spikes glycoprotein will lead to bind with the CD4 because it is a specialized for the binding of the CD4 just. So after the binding with the CD4, so here is the binding will be occur, and in this way you can see, and that will be fusion with membrane. After the fusion, here I am drawing just for understanding, so the fusion will become a endosome, so the endosome formation will be occur, and in this way the endosome is basically this diagram is basically confused, but you can understand that the infolding of the membrane will wrapping around to become an endosome, and this endosome will be open into the cytoplasm. So inside is the cytoplasm. So this is the membrane of the CD4 T lymphocyte. So the CD4 is helping the immune response. So the T helper cell is it is also known. So that during the fusion we can inhibit by the in infuvi infuviri infuvertide. This is a medicine which we can stop it. And on the other hand, here is the maraviroc. This is a medicine can inhibit by the binding site. So this binding site can be inhibited by this drug. But anyhow, when entry will be occur, so the RNA containing virus will be a replicate. So is in this way the reverse transcriptase enzyme begin to reverse transcription of RNA into the DNA double helix, and this double helix structure of the DNA will again the helicase enzyme used to again replicate in this manner. But in this way we will just understand the reverse transcription. After the reverse transcription from 3 prime to 5 prime to, so here is a 3 to 5 prime is the antiparallel strand. So in this way you can see, and this is the reverse transcriptase enzyme used for it. Okay, here is a DNA double helix filled with form after this reverse transcription. So this reverse transcription will become a DNA double helix. So the helical structure will be occur, and this helicase enzyme can be used to again produce the RNA. So this RNA also contain a reverse transcriptase enzyme binding for the another packaging for the exocytosis of the new mature HIV virus. So in this way, the rap helicase enzyme used for the increasing of the copies of the RNA of the virus. So that is important things. So yeah, you can understand from this. But now we will discuss about the nucleus and what is the function into the nucleus for this DNA double helical structure. After the becoming a DNA double helix, we go into the nuclear from the nuclear pore into the nucleus. So inside of the nucleus of the CD4 cell contain their own DNA, and their own DNA is basically the integrase enzyme is also come from the virus, and that's bind with the DNA and move into the transporting protein to move into the nucleus. After the movement into the nucleus, LTR to LTR will be uh, end of the segment. So the ligase enzyme and endonuclease enzyme used for this activity, and the integration and integrase plus ligase enzyme used for it. And after this binding with the DNA of the host, so this information will be filled into the nucleus in the human nucleus. So basically after the binding and contain this information, which that is the viral information, and after this transcription will become a produce messenger RNA, and that messenger RNA can be go out. So this messenger RNA is replicate from the viral DNA, viral genome region. So the viral genome region after the move out. So the endoplasmic reticulum. This endoplasmic reticulum will transport the messenger RNA, and after the machinery of the protein synthesis, the ribosome will be bind within it, and in this way the messenger RNA will translate into the protein, and that protein is important for the packaging of the virus. So in this way the RNA will be packaged wrapping around through a Golgi body using, so the machinery of the host can be used for this function. So that is why the virus is important, and it can be used the machinery of the host. So the receptor is important, CD4 receptor, and that's without CD4 receptor the virus cannot go into the cell. So after this, packaging will be occur and the Golgi bodies will be modified this protein, and that is the latent provirus will be occur, and this latent provirus will become a immature virion, and this immature virion also contain glycoprotein, reverse transcription. So all this activity basically is the proteolytic cleavage will be occur, and after this move out is known as the exocytosis. After the exocytosis, the rupturing of the plasma membrane of the CD4 will damage the CD4. In this way, the immune response will not occur. It means the helping for the immune response will not occur due to the damaging of the CD4. So the death of the CD4 due to the a lot of virus will be produced, and this a lot of virus will damage the DNA of the CD4 as well as the damaging itself the cell of the CD4. So the CD4 will be damaged. So in this way, the recruitment of innate response and the adaptive immune response will not occur, and the all disease can be occur. So the proteolytic cleavage can be inhibited by the lopinavir. So the here is the mature virion. So in this way you can see and understand that life cycle of HIV virus. So in this way basically is that many disease can be caused because the infection sensitive our body will be due to the loss of the immunity. So the every disease, for example influenza virus or other problem can lead to proceed more dangerous. So thanks.